Hello, everyone. Welcome to our next installment of my little biotech talks, TED Talks. We are going to be moving away from talking about organelles to specifically talking about the plasma membrane, a structure that's very important with maintaining the stability and functioning and communication of cells. What we're going to be talking about are what makes up the cell membrane, what parts of it does it have, what other little gadgets it has, and what all their functions are going to be. So let's begin. So the basic building block of a plasma membrane, the bricks that make this thing work are the phospholipids. The phospholipids. Throwback to macromolecules. Phospholipids have two really cool features. One, it has a hydrophobic tail, which means it hates water. And then it's going to have a hydrophilic head, which loves water. Some other key identifying features of the phospholipid are its phosphate group head, right here, phosphate head, and its glycerol, right here, glycerol. Phospholipids also have two tails, one of which is saturated, meaning there is no double bonds, and it's going to be a straight line. And then we're going to have the unsaturated tail, which is going to be bent and kind of kinky. And this is going to be because of it having less carbon bonds. It has double bonds that bend the molecule. And I'm going to write only single bonds for the saturated tail. Phospholipids, due to their nature being having a hydrophilic and a hydrophobic section, this makes them amphipathic, meaning that they have hydrophobic and hydrophilic parts. The phospholipid is going to make a bilayer, two layers, one, two layers of phospholipids. Phospholipid bilayer. And the main point of this phospholipid bilayer is to separate the internal cell environment from the external environment. So keeping everything on the inside, and keeping everything that's supposed to be on the outside. The plasma membrane is what we call selectively permeable. Key vocab right here. Selectively permeable, which means things can come in and out. However, go in, go out of the cell. Things can go in and out. However, this plasma membrane acts as a bouncer, and it has a list of who can come in and who can come out. And so this plasma membrane is going to act as a bouncer, regulating what comes in and what comes out. And so it's again important to note that the hydrophilic heads will always face towards the watery environment. They will always face water, and the hydrophobic tails will always face away from watery environments. If we were to chuck these phospholipids into water, they would eventually orient themselves into little pods, keeping the hydrophilic sections on the inside and the hydrophilic things on the outside and the hydrophobic on the inside. Head, tail. On top of calling it selectively permeable, we also call it a fluid mosaic model, which means that, well, let's break it down. The fluid part means that this membrane is held together by weak bonds, and so therefore all these little molecules, all these little phospholipids can move around. They can go this way, they can go this way, they can go this way. etc. It's like they're at a concert and they're all jam-packed. Like how I like to think about this is, let's take a pool of water. We have a pool of water and we throw all of the ball pit balls from McDonald's into the water. And all those balls are going to float on the top of this pool that we got right here. They're going to float on the top of this pool and they'll be able to move around as they please. They'll still be tightly packed, but they can still move around. What determines their fluidity is temperature. When it gets too hot, it starts to denature and break apart. And when it gets too cold, it starts to slow down and become more solid. We do not want a solid phospholipid bilayer. We do not want a solid cell membrane. It's meant to be fluid. And luckily, we have some things that protect it from getting too solid or becoming too cold. And so we have this thing called unsaturated carbon tails, hydrocarbon tails, the kinky tails that we were talking about. Right here, right here, right here. The fact that these are bent means that they do not become solid at room temperature, just like oil, just like um, warm fat. They do not become solid at room temperature. And so again, like these kinky tails, they prevent this from happening. They prevent the tails from stacking on themselves in a straight matter. They prevent this stacking happening. This is what the structure of butter looks like at room temperature. Butter is solid at room temperature, and we do not want our phospholipid bilayers or plasma membranes to be solid at room temperature. And these kinky tails, these bent tails, keep it from doing that. Another thing that's going to help maintain its fluidity is cholesterol. Ooh, what do I see? I see 4 carbon rings right here. 4 carbon rings. Throwback to steroids. Steroids are also a type of lipid. They help with maintaining the fluidity at high and low temperatures. And they also help with preventing phospholipids from packing too tightly. Okay, so we talked about the fluid part. Now the mosaic part. What I mean by mosaic is that think of like a mosaic painting. It's a bunch of different tiles, a bunch of different pieces of glass, etc. And so on the phospholipid bilayer, we're going to see a bunch of different things on the surface. And we're going to talk about all these different things, all these different macromolecules that we're going to see on the surface of our phospholipid bilayer. Our first part is that we're going to talk about the proteins. And so we have 2 major types of proteins. We have integral proteins, and these proteins are embedded, keyword, embedded into the phospholipid bilayer, which means that they are transmembrane. They go across both membranes, and they are also amphipathic, meaning that they have hydrophilic and hydrophobic sections. So I'm going to zoom in on an integral protein. Go away, keyboard. Oh, what did I do? Oops. So we're going to zoom in on an integral protein. So I'm going to color all the hydrophilic sections in, let's say green. That's a better color. In green. So the top and the bottom, hydrophilic. And then I'm going to highlight all of the hydrophobic sections in red. And so all the hydrophobic sections are going to be in the middle, just like the tails. And then our second type of protein is going to be the peripheral proteins. Peripheral proteins. Peripheral means that they're just on the side, they're laying on the top or on the bottom of the phospholipid bilayer. They're not bounded too tightly and they're able to move around. And we'll see one right here. Let's do a different color. Let's do red. Highlight in red. Phospholipid, peripheral protein. And what these proteins do is help with bringing in different types of substances or taking out different substances. And they can act as bridges or channels that allow things to come in or out. Next, we're going to talk about the membrane carbohydrates. So we have two types of membrane carbohydrates. We have glycolipids and glycoproteins. Glycolipids and glycoproteins. When you see the word glyco, we mean carbohydrate, and then the rest can speak for itself. Lipid means lipid, protein means protein. And these primarily help with cell communication. Function is cell communication. My hand. And we'll be talking about this in more detail in a later unit. But just know that these function for cell communication. And so all of this was for plants, for animal cells. This is still going to include plant cells to some extent, but now we're going to start talking about plant cells for just a little bit. The plant cells have an extra layer of protection called the cell wall. And the cell wall is going to go around the plasma membrane. What the cell wall does is that it gives it shape and structure, it gives it protection, and also acts as a way of regulating water intake, how much water is coming in and out of the cells. How they are made up of cellulose, and they are thicker than plasma membranes. And the way that these cell walls that these plant cells communicate is through their plasmodesmata. Think of the plasmodesmata as little openings. So let's do this in red. Little openings that allow for communication between cells. Little highways, little doorways that allow for communication and nutrients to flow into plant cells. But again, we'll talk more about this type of communication later in the year. Well, thank you for listening. We will have another section in just a moment. We'll be covering the membrane transport and facilitated diffusion in the next section on our next Ed puzzle. So I'll see you in a bit.